Hey, science learners, welcome to Wednesday's lesson. And today we're going to do the experiment or the dissection, rather, of a seed for why are there so many plants. So we're going to start out with your table of contents page. Today should be the 16th. That's right, the 16th. And the title, Why Are There So Many Plants? That's page 7. And then tomorrow we're going to do another one. So I actually got both of them written there. But you really need to worry about 9:16. I'm going to hold it up close, let you pause the video right here. Why are there so many plants? So pause, update your table of contents page, please. And then flip to page 7. If you're back with me, we need to set up page 7. At the title at the top, why are there so many plants? This is our page 7. So you see my 7 right down in there in the corner. And then it's got a blank space. We're going to do a little bit of scientific sketching today. And then below it, our class claim and our class evidence. So pause the video. Why are there so many plants? Class claim and class evidence on page 7. Go ahead and pause. All right, back with me. All right, so if I held up a picture of an ecosystem, and this is a picture I have of a riverbank ecosystem. Do you see it? One thing you should notice is all the animals, of course, and you can name some of the animals if you'd like. All the animals. But you also see all the plants. And if you counted, you'd have tons more plants than animals. So why are there so many plants? Well, there are so many plants because there's so many things that need plants for that energy transfer, right? Things that eat plants like herbivores or omnivores who eat plants and animals, right? But plants are also awesome at reproducing. And we just finished a quiz when it talked about structures for reproduction. And that is, in plants, is the seed. The seed is awesome. It's the best at reproduction, and they can do it so fast. They can grow lots of copies of themselves with very little errors in them. It's not like animals, right, that takes a long time. Like humans, humans not only take 9 months to develop and grow, but then to raise to a full adult size, it takes many, 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 many years of constant care, where plants, they don't have that energy, but they're using to do that. So today we're going to be looking at a lima bean seed. And what I need you to do, if you don't have a lima bean seed, you can do any other seed that's big. You can use any kind of beans. Beans in particular are pretty good at it. But what I had to do is I had to soak it. And lima beans work so well because they're big. I soak it overnight so that I can take it apart. And so you're going to need that. You're going to need a magnifying glass. Like I said at the beginning of the year, if you don't have a magnifying glass, you can make one out of a glass of water that you can see through. So what are we trying to see in our seed? All right. I want us to see what is in there in person. But in particular, you should be looking for 3 things. All right. You should be looking for the embryo, which is a tiny baby plant ready to grow. And it looks white, and it looks like it's got a little bitty stem, and it's got a little bitty white leaves on it. And then you should look for the inside, the nutrient part. If you read your chapter, you know what I'm talking about, the nutrient source. And that's the cotyledon. It's a weird science word that I just love to say because it sounds awesome. Cotyledon. And then on the outside of the seed, on the outside right here, you have the seed coat, which is its protective cover, which was also in your chapter that you just read for me on Pearson Realized, right? All right, so we're looking for those 3 things today. So we're going to take apart your seed, and then we're going to scientifically sketch it into your journal. Okay, so I'm going to take apart my seed, and the first thing I'm going to peel off is the seed coat. Do you see it? Do you see the seed coat? My seed coat's coming off really well. But then again, I soaked my bean like 24 hours at least to get it to do. And it's a big bean. So this is the seed coat. And I'm going to use my sensory observation. Yeah, I can see how it feels. It looks white with lines in it. It feels smooth to the touch, slightly sticky. Ooh, it's got a yeasty smell to it. That's an unusual smell. I'm not going to taste it because we don't do that in science. It doesn't really make any noises. It's just sort of slick. All right, and that's my seed coat. And I'm going to pull apart. And this one, look, this one, look. Do you see the embryo at the little baby leaf? You see the embryo right there on the end? This is my embryo, and that's a little baby leaf right there. And then this part, the other half of it, that's the cotyledon. That's what it uses as fertilizer to grow as it develops. So we have seen all three parts of the seed. So now it's time to do a little scientific sketching in our journal. So I'm going to get a pencil. And in my journal, I'm going to do the best that I can. Remember to do the A, B, C, D, and E. Accurate, big, colorful, descriptive, detailed, and explained. So I'm going to start out and draw my bean to the best of my ability. Okay. And then I'm going to draw my seed coat on there. And my seed coat had stripes that I saw. I don't know if you can see it as well as I can. Stripes that I saw. No. On there. And so I'm going to try my hardest because scientific sketching is one of those things. The more you do, the better you get, all right? And so I'm going to try all my steps for scientific sketching. I'm going to do the seed coat. Then I'm going to do the embryo, which looked like a little tail. And then it had a little bitty leaf, baby leaf that was not even green yet. That was on the end. And then my cotyledon, which was behind it, right? So I'm going to draw the other side and make that my cotyledon that matched there. All right. And so I got to do colorful now. Mine was more white and yellowish colored. So I'm going to get my colors from my colored pencils. And I'm going to color in those cotyledon. I'm going to get my white and my yellow colored pencils because the cotyledon was a darker yellowy color than the seed coat. So I'm going to color that in. And the embryo was the same color as the cotyledon. So I'm going to color that the same color as the cotyledon. And then my seed coat was whitish. So I'm just going to go over it with a white colored pencil. And it won't look that different, but I'll know that it is done colorfully like you should. Okay. All right, and now I'm going to label each part as part of my explained. Remember, there are three parts. The embryo, the seed coat. Uh-oh, I wrote the wrong word. The seed coat. The embryo. And that long word, cotyledon. You know what I'm going to do for you? I am going to hold up my reference so you can see. Oh, see, I got all three labeled. I'm going to hold up my reference so you can see it. And that way you can pause the video and get the spellings and the words right. All right, so there we go. The embryo is the tiny plant inside. We saw it. The cotyledon is the bigger part of the seed, the inside of the seed that the plant uses as food. The seed uses as food to grow. And then the outside covering is the seed coat, and it keeps the insides of the seed safe. So there we go. So why are there so many plants? What's our claim? I think there are so many plants because plants are good at reproducing. What did we do in our experiment to show how good plants are at reproducing? You can pause it. What did we do? I took apart a seed to see how it works at reproduction. All right. I'll let you pause here, and that'll finish our experiment up. I hope you have fun dissecting a seed. You can take a look at it with your hand lens. I hope you see all the 3 parts that I need you to see, okay? And I hope to see you again soon. Be safe.